Right, so we are now recording. First off, while we wait a little bit, sometimes folks trickle in. Does anyone have any questions um, regarding, well, anything, right? If you have questions about the study guide, it should be coming up soon, hopefully by tonight, if not early tomorrow morning. It will be a page in Canvas. Right, there's a question. Good. Where will the question be posted? There's two places you can find it. You can find it in the same Zoom tab on Canvas. You can also find it in the Panopto or Panopto or Panopto um, folder, which I believe you guys also have access to. But I would go to that Zoom tab and look under recordings. It should be there. Usually takes Zoom about 10 minutes or so to do an hour recording. The last period, we did stay the whole hour. And part of it had to do with questions that some students had, which is the main point of today, to sort of address your questions. Right, everyone should have the key to that quantitative analysis sort of handout, or most of the key. The rest of it was left out because they should be hopefully um, dimensional analysis or just problems that involve units, right? Different units, changing them from like kilograms to pounds and so on. The other thing that came up previously, as we wait for more people to sort of come on in, um, so we went and we talked about, that's escaping my mind. Oh, will we have all the stuff that's in the syllabus in test one? That is the goal. I believe it's an attainable goal, especially since the IV fluids PowerPoint has a video now that goes to it. So I may not go over that in detail or a lot in class, but there is a video that's connected to that. And part of the reason for that has to do with this quantitative analysis worksheet. In the video, I go over the sort of definition or try to clarify some of the things that I heard and that were questioned about the quantitative assignment regarding concentrations. What is a mole? How does that mole relate to osmolar concentrations and things of that nature? So it's not just IV fluids, but it's also a little bit of that. Additionally, for the study guide, this IV fluid presentation would be your go-to place for like I don't know, studying um, in terms of questions that may come up on the exam regarding fluids or IV fluids. You would have them, or your answers or your questions would come from that PowerPoint presentation. For example, one would be, what is the difference between, uh, thank you for those of you that are transcribing the meeting. I forgot to click that on. Um, uh, so an example of a question regarding the fluids would be, what's the difference between a colloid and a crystalloid IV fluid, right? Um, there's a question, how to study for the quiz or quizzes. So your quizzes are basically open notes. Just about everything is open notes. The exams, the in-class exams or quizzes, those are not necessarily, I mean, you get a sheet of notes, but not as many open notes as you would if you were taking an online quiz. So how to study for these, I would recommend the PowerPoints. Your OpenStax book is okay, but as I've said before, it has a lot of um, topics that it kind of doesn't get into the detail that we get into the PowerPoints. For example, case in point, the protein receptors that transduce signals into the cells or signal proteins and transduction cascade mechanisms that we will talk about in lecture tomorrow. So I would focus on what we go over in lecture, PowerPoints, and then go from there. Remember, none of the papers, unless otherwise stated in advance, none of those will be on your exams. However, for the points that you're going to get for these meetings, right, when you do your summary, you may see a question or two regarding one or a couple of the papers that we go over or that were assigned. So with that said, I believe we have a, well, 50% turnout or so, which is what I kind of expected. There is an agenda or a little sort of protocol or the sort of goals here, right? So we're going to go over lecture topics briefly, lab topics, yeah, up to the 5th week, not just 4th week, but 5th week, just to give you a heads up. The main idea there is to try to read your labs before the actual lab day, or at least leaf through them. And then journal articles, we'll go over those two briefly, and then any other questions you all may have. So with that said, if you don't have any questions, I don't see any new questions on the chat, then we will move on to the lecture topics. And so the idea of this is for me not to have these topics anymore, but eventually to have us or you all come up with, okay, what have we been talking about in lecture in the past, I don't know, a few weeks prior to this meeting, right? So that's the idea beyond this, but I'm giving you kind of like a little bit of a template, if you will. So these are the topics, right, in the following, or in this week, few weeks that have passed in the following weeks. So we have concepts, homeostasis, chemistry, proteins, protein signal transduction, and you can read the rest. So one of the things I want to ask you all is, what is one or two, maybe if you can think of two, right, concepts that we've talked about, like a very core or general concept we've talked about regarding physiology. I'll give you one. I'll give you like the most umbrella concept, and then from there you guys can sort of pick. So that one of the most sort of universal concepts in physiology is it's that it's an integrative science, meaning it just like many other branches of biology, it uses things like chemistry, biochem, you name it, to sort of explain or convey particular mechanisms and their understanding. So it's not just a sort of one subject matter does it all, but it actually brings in a whole bunch of perspectives from different branches. So that is one huge concept. Hence, we discuss chemistry and things of that nature. So with that said, can anyone sort of either state, put in the chat or anything like that, what could be other two concepts of physiology that we've covered? Right, we have.
I guess you mentioned form and function is like a concept of physiology.
Okay, so form and function, and we had anatomies in the chat also. Okay, so those are important concepts.
It's a mechanistic approach.
Yeah, mechanistic approach. And that leads us to essentially what you have under concepts, right? I didn't mean to put them as such, but underlining those or putting all those together, like anatomy, form, and function, in terms of physiology, is the understanding of homeostasis, and with that, the chemistry involved in homeostasis. So those are two big concepts, right? Big, like universal ones. Yep, cell-to-cell communication is another concept, but just to reiterate, um, the sort of big picture concepts here would be homeostasis and the chemistry we've been talking about. So let's expand on these a little bit. What about homeostasis is integral, or makes it a sort of huge, wide concept in physiology?
Homeostasis is what allows us to exist. It's what keeps us at that set point or within the range of that set point so that we can be alive and function in the way that we do.
Great. Yep. Feedback loops I have in the chat as well. Dynamic equilibrium. Oh, I like that one. Cool. So a balancing act, right? I like the fact that the word dynamic was before equilibrium, not just equilibrium. So yeah, this idea of regulation, of maintaining a set point that each individual or each system is in tune with in order to stay alive or perpetuate its activity, right? So with that said, how does chemistry or what huge concept in chemistry or idea, we can even say that, connects with the idea of regulation, feedback loops, and all the other items you guys talked about that are pertinent to homeostasis? What about chemistry makes it important to this pot here?
Everything from like the polarity of different things like salt and electrolytes and water balance to um I lost my train of thought, but like to start just having things that are polar can explain how fluids move in between the body.
Right. Then good. That is the one of the main ideas here. We have shaped molecules or attractions, and whether they repel, correct, electrochemical gradient signaling. Uh, Indeed, right. All of these are the outcomes of what chemistry provides to these homeostatic or the homeostasis concepts, which is basically that's the way these systems communicate. They communicate through chemistry, right? You cannot have or have a particular adjustment in pH without the chemistry behind it. You cannot have ideally the adjustment in temperature without the chemistry that leads you to that. So they're interwoven, if you will. Uh, One sustains that communication factor or the basis of the communication, and the other one is the effects of that. So you can't really sort of not convey physiology, homeostasis within that without addressing the chemical aspect of this. And to that degree, proteins is right underneath, right? Because part of this chemistry we've been talking about is, yes, the polarity, how molecules interact with each other, but the sort of layering or the hierarchy of items. And in that perspective, proteins are sort of at the center point of all this and the formation of proteins. Hence, we talked about the primary, secondary, tertiary, quaternary protein shapes. And one of the questions we got in the previous period was basically, I kind of don't understand, or it's not very clear to me, the importance or why primary has that shape, why secondary has those shapes, and why does hemoglobin, why is it affected right by any of those shapes or even the primary? So it was a very interesting question because it was pretty straightforward, and it addressed or was focused at the core of understanding one, that form and function concept we talked about, right? And two, the chemistry behind this, and you can even add homeostatic controls and whatnot. But for now, those two. So what I mean by form and function is even at this level of this molecular level of proteins, primary through quaternary shapes, uh, the form of these proteins or how they are shaped and can contribute or does contribute to a particular functionality. And so one of the things we discussed was, well, how is that so, right? How does that sort of, how does that become an outcome of the shape of a protein? And so we talked about essentially because I'm in a room that's pretty cold and I was wearing a beanie, not anymore because I was running up and down the stairs a while back. Anyways, and so it dawned on me that the beanie, right? If you look at or any knit hat, the string that makes up the hat, that's like your primary protein. Without those strings, you won't have essentially the shapes or the ridges of the beanie or the particular pattern of the beanie. And so that leads us to the idea of the secondary structures, the alpha helices and the beta sheets. Those promote particular patterns, right, on certain proteins based off of those primary proteins. Now, we kind of slowed it down here a little bit and got into the idea, well, how is it that the primary proteins form these alpha helices and beta pleated sheets? And in our perspective, or for all intended purposes for our class, the idea behind this is the fact that these molecules are in 3 dimension, right? They're 3-dimensional shapes. And as such, there's going to be times where the proteins, we're talking about the primary shape, will fold. The longer it grows, the more it can fold in the environment. It's in an aqueous environment. So the longer it folds, the more, or the longer it is, the more it can fold. As it folds, it creates these sort of pockets or little cavities known as domains that are specific to that certain area. And within these domains, you could potentially link or connect the protein via its R groups or functional groups of the amino acids that make it, and then, maintain the shape or promote another shape. So in other words, to a certain degree, the formation of the alpha helix and the beta pleated sheet has a lot to do with environmental conditions and the stability of the protein at a given size. Remember that a peptide, we said we defined in class as anything being 2 amino acids or 50 amino acids long. Beyond that, we will refer to it as a polypeptide. Even though something that's 50 amino acids is in the nature of polypeptides refer to proteins that are 50 plus amino acids long. So from this idea, right, of this beanie, we said, okay, well, the ridges or whatever other shapes that beanies have, a beanie hat may have, has to do with these accumulation of secondary proteins. And recall that in secondary proteins, it could be that you have only alpha helices or beta pleated sheets or a combination of both. And then when you get to the larger proteins or the tertiaries, you start having these subunits of those proteins, which connect in space and form, again, more domains. Larger proteins may mismatch or misconnect certain areas and then form again or refold. And then lastly, the quaternary one is essentially related to these even larger proteins, which we refer to as either more globular or globular proteins like hemoglobin, where you have many more subunits. Hemoglobin has 4, right? 2 alpha, 2 beta. And so this constitutes that idea of scaffolding particular shapes. Now, a question that you may have in the exam would be something like, can a quaternary protein have or potentially have an alpha helix or a beta pleated sheet within its structure? And the answer is yes, you could. Again, you may not have that throughout the whole big protein, but there's certain parts that support that protein that stayed in that shape. And that was one of the ideas behind that, or one of the discussions we had. Do we remember what allosteric effects are? Allosteric.
They non-inhibiting. What allosteric is that? Something would change the enzyme shape to prevent it from connecting to a substrate.
Right. So allosteric effects are the effects that cause the conformational change on the binding side of an enzyme, either directly or indirectly, right? And these changes can either destabilize the enzyme or stabilize it, depending on what it is. Hemoglobin is used a lot to look at allostery, but remember, hemoglobin is not an enzyme, it's a protein. But like an enzyme, it has a binding site. That binding site can be affected by allosteric events or allostery. So if it's a competitive inhibitor, that means that particular protein will bind into the binding site of the enzyme or a protein and thereby block the substrate that's supposed to go there, right? So that's one. And then that's competitive. And then competitive, right, is when, or non-competitive is when a particular molecule or something happens that affects an area or a region or the a particular domain of a protein other than the binding site. However, this issue on any other place on the protein or enzyme does then affect the binding site, and the substrate may not be able to bind, or there may be another issue. And along the same idea of allostery is this cooperation or cooperative effects, which is an allosteric effect that some enzymes and proteins share based on the amount of substrate that's there. For example, hemoglobin will go through this, right? I said in class, when oxygen is in abundance, the subunits of hemoglobin will go through this allosteric effect of cooperation or cooperativity and promote the binding of hemoglobin. And in general, because we're going to get into more detail later on, but in general, this happens when oxygen is abundant or needs to be transported from one part of the body to another. So that's what's referred to this, and this is what allows the finite regulation of particular enzymes or inhibition of certain mechanisms via this pathway or via these pathways. And so this is important in understanding the next thing we'll talk about in class, which is protein signal transduction. Before we get to that, or actually, let's just make the connection here. So proteins that are on the surface of a cell are receptor proteins for the most part. Some of these are channel, but they receive a signal, right? And then the signal, once it binds the binding sites, changes the conformation of that protein, and then a whole bunch of events happen after that. So that's kind of like the idea that we sort of piggybacked on in terms of allostery. Now this is in line with the fact of what protein signal transduction does. And so one question I have for you all is what is signal and what is transduction? So I'll ask you this. Everyone that's in the Zoom right now is receiving a signal, right? You can hear me and we can see the lecture topics slide, correct? Or think about that. You don't have to affirm it, but if you said correct, yes. So that is a signal. Everyone's getting the signal, right? But there are 19 of you. There's 19 of you. So that means the signal's being spread, and that is the transduction process of this whole event of signal transduction. So if there was no transduction or spread of the signal, then only one of you, be it Victoria, Sean, or any of you, would be the only ones receiving the signal. And the rest of you, which would be part of the cell, would be like, okay, what's going on? So proteins, especially these signal transduction proteins, like ion ligated, and the G protein coupled receptors, those play an important role in spreading the signal in a cell. The reason for that is because if you recall from lecture, we said that the cell is like a whole new world inside like a system. So one area of the cell from point A to point B may be eons or distances away. So to get messages across, you need a very efficient, fast-moving, fast-spreading mechanism. And so this is where these proteins come into place or into play. The other reason they're used a lot is because a lot of the signaling molecules cannot pass the membrane, so they need a protein receptor. And we'll get into that more in detail later when we talk about hormones. So that's why we are looking at protein and signal transduction in the next. Let's see. So would it be like being? Yeah, that's a good analogy. So there's an analogy here about beating the heart and how the brain signals the SA node, and then the SA node has to transduce that signal to other fibers in the AV node, and then those fibers have to spread the signal to bundle fibers. I would even extend this to the fact that at the SA node or within the atria, you have autorhythmic cells. So they themselves act as the spreaders of information and then promote the signaling. So it's that's the systemic, that could be a systemic view of what we're talking about. In this case, as you'll see tomorrow, hopefully in lecture, it's it's within that enclosed system that cells are, right? So that sort of the idea of compartment and compartmentalization that we talked about, that sort of um, requires a different approach in terms of signaling for the most part. And part of the reason for this sort of um, this concept of signal transduction has to do with what you all said earlier about homeostasis and how the cells need to move things, or need to move, are affected by electrolytes and whatnot. And so two things that keep cells going, keep them alive, are managing electrolyte balance, just like we all do with water and drinking electrolytes, or having electrolytes in our body, and also the digestive section. Obviously not the eating of food, but more so the um, use of substrates like glucose, in some cases proteins and fatty acids, to help promote the formation of energy. So in other words, you cannot balance electrolytes using proteins and gradients or forming gradients without having a source of energy. And you can't have a source of energy unless you have the means and the methods by which you, as a cell, can bring that in, break it up through cellular respiration and its mechanisms, and then use it. So without all this, right, everything sort of kind of collapses. And so it's this becomes a very efficient process through the cell signaling we talked about. And to a certain degree, this signal transduction helps accentuate or make these two general mechanisms of electro balance and energy um, acquisition as efficient as possible for the life of the cell. So we're going to get into that hopefully this week, especially electrolyte balance and protein transduction. Questions before we move on to your labs? You can put them in the chat, or you can unmute yourself. All right. I don't see any. You can always, bring them up in lecture. So Tuesday, we're doing a continuation of homeostasis, but it has to exercise. One of them, you sort of come up with some kind of stimulus to increase heart rate or whatnot. And the other part of that is something called it's a reflex that we're using. Oops, there's the question on the chat. Let's see. In the future, can we watch? So great question. How do you get points for this? Well, just by showing up, you won't really get points. However, if you're taking some notes or you are at least it's kind of in and out of the particular things we're discussing, you may not need to view the video. The video will be available in the same Zoom tab shortly after this recording, and or the Panopto folder. So there will be a page, hopefully by Tuesday, you'll get a whole week to do it, where you put in your summary of what you got from the sort of discussion or meeting. And there may be a question or two that's going to be related to the readings that we were supposed to do that week, more so the papers, as we'll see in a moment. But I want to give you a quick heads up. Let me just take a look at the time here. Okay, we have about half an hour. Quick heads up of the lab, especially tomorrow's lab. So let's go ahead and check it out. I'm going to share it with you all. When I upload this presentation, if we need it or whatnot, I'm still going to upload it, but you may not have the ability to click on the same things I clicked and get the link or be linked to the lab, because those links or those hyperlinks are to my computer. They're not necessarily to to the labs in the web. But you have access to the to these, sorry, except the last one that I'll show you. So again, homeostasis, the sort of loop mechanism behind homeostasis, and then an example of what's going on with heart rate, right? Why are we picking heart rate, or why is heart rate so indicative of some kind of homeostatic control? You have to design a brief experiment, right, in which you're going to alter or promote or stimulate the alteration of heart rate amongst your subjects. And we're going to use this thing you see here, which is the sort of sensor. It's going to be connected to the MacBooks or any other computer. And you basically hold those, right? You hold that sensor and it'll test your heart rate. The idea here, though, is to come up with a design of how to stimulate heart rate. Now, if you pick an exercise, make sure, again, you are cognizant of the fact that strenuous exercise, especially the night lab, if we're not hydrated or have not been hydrating as well as possible, may lead you to serious issues like the one student that unfortunately got rhabdomyolysis. So do everything based on your limits. If you believe that your lab group has selected something you can't participate in, you can always opt out. I'll give you a big sort of clue here. A lot of people pick, or some people pick something like a scary video. But again, all of this as a stimulus, right? All of this, keep in mind like safety and everyone's overall health. So once you design the experiment, you have a table, right, in terms of how to um, um, sort of obtain the data. And then one thing we're looking for also is recovery time. In other words, you stimulate the person, or you stimulate an increase in heart rate, or what you believe would be a stimulus for increase in heart rate, take the measurement, and then time the person in intervals to see how long it takes the person to go back to their initial resting rate or basal heart rate, right? So let's say if my basal heart rate was 66 beats per minute, fine, put that in there. You might want to measure it 3 times at least and get the average. That's just the way I would do it. And then from there, after the stimulus, quickly measure the heart rate, and then do intervals and see how long it takes me to go back to 66. Now, this is where you have to make sure you pick, or you talk amongst each other and select an interval of measurement that may, catch or can best catch that sort of um, uh, time of recovery, right? And it says recovery of heart rate within 5%, which means, it gives you a little bit of buffer room there, because most of us will not go back to exactly the same um, resting heart rate or basal heart rate. So that is one exercise with some questions, of course. Then the next exercise, a little bit more involving, a little bit more confusing. It's the cold pressor test. It's like a little reflex that we're testing. It's basically where you dip either a finger or um, the hand in various temperatures of water, and then you have a pulse ox on the other hand, and you see whether or not you are more prone to um, the stimulus on the right hand versus the left hand. And typically most right-handed people are more prone on the right side, and left-handed people on the left side. But regardless of which hand you're more prone to, or could be more prone to, eventually acclimation will sort of even this out. So you're given a table of when to measure this in terms of seconds. You can do up to 5 participants, but I would say at least 3 would be fine. And the idea here is to see what's going on in terms of the data. This is just raw data. Later, hopefully, when you use your this for your homeostasis lab report, you can use the raw data to make some graphs and talk about the outcomes. So that is lab. What is it? Lab, the next lab, tomorrow's lab. And then the following week, which is week 4, is a very busy lab. It's actually a fun lab, but there's just sometimes way too much going on. Let me show you. So that is the basically, excuse me, the cell lab / osmosis lab. There's there's 3 exercises here. Of those 3, um, let me share the lab with you. The most involving one is the second one. So heads up on these labs, or many of the labs from here on, you don't have to follow that particular order, especially if you're doing division of duties, right? Or division of um of exercises, like if you're splitting up the work. So what I would do here is start with the second lab. I'm going to go first, sorry, the second exercise. I'm going to go with the first one first. We'll talk about the first one first. The second one involves making cells out of cellulose paper. You need to make sure you dip the cellulose paper in water first so that it becomes loose enough for you to make the cells within these particular strings, right, tying off the ends. So we'll go over this briefly in class as well. Main point of this is on week 4, try to get a little bit of the reading prior to coming in lab. So this is one that can work very nicely, but it is sometimes very difficult to do for one reason or another. So the mad phase means if you make a well and it's that deep where it hits the bottom of the Petri plate, it's not going to work because the dye will leak on the bottom. So you want a well that just barely hovers over the surface of the plate. How do you make the well? Well, no pun intended, I used the word well again. We use a small piece of, what's it called, of a straw. And we wedge the area, right? We make the mark on the area and you have to wedge it back and forth gently to loosen that area, and then use like a little toothpick to dig it out very carefully. And eventually it'll look something like this. You can use 1 Petri plate. Every table will get 2. I would say use 1 and split it down the middle like we have here. You can draw that line on the cover part, but just make sure you don't move it as you're studying your diffusion. And the idea here is to see which dye will diffuse faster in this medium or this agar. Normally the dye that is more towards the nonpolar, smaller side of things will diffuse faster. How do you quantify that? Well, you measure the diameter of the circle that is formed. All right. This is the one that takes a long time. This is the exercise to forming your cells, right? Or bags of cells. So you're going to use water that has sugar in it and a dye in it and salt. And the idea is each of these cells will have that concentration or it will be in the environment. We're trying to see what's permeable across the bag, or what can diffuse across the bag, either salt, the dye, or the sucrose. So the confusing part of this is how long do you leave them in the beakers for? What should the beakers have and things of that nature? My recommendation is look at the table, because the table literally explains it a little bit more clearly than the words as to what should be in the beaker. So you have a beaker with deionized water, beaker with 40% glucose solution, a beaker, another beaker with deionized water, and a 4th beaker with deionized water. So you can set up your beakers, and then you have contents of the bag, right? So you set this up. Before you put the bags in the beakers, you have to weigh them because the weight may change. And then you test certain solutions. Not all. You see the darkened out blocks, you don't do those, but you do the solutions that are stated here or on the experiments that you ran in the beakers. All right, and so the last one here that you can be doing while you're waiting for your beakers or whatnot is, remember, that experiment needs to, or that exercise needs to be run for an hour, is the sheep's blood and the use of different concentrations of solution in the slide or the blood that you're going to prepare, or the slide you're going to prepare using the sheep's blood. My biggest recommendation here is first look at the sheep's blood without any solution that you would add or are going to add to it. So in other words, just make a slide of sheep's blood, check out the relative size of the cells, right? Get a either picture or a mental note of it. Then you can add the solutions. Just be wary that if you add the hypoosmotic or hypotonic solution and you take too long to look at the cells, you might promote the lysis of the cells and not see anything. So keep that in mind. So that's basically it. Now, last but not least, just a brief, this is not on your canvas, but just a brief heads up on week 5, or the slide for week 5. Let me go back to the sharing here. And if you have any questions while I'm going through this stuff, by all means, go ahead and put it in the chat. Get it here. Right, so make sure I'm sharing that. Yep. All right, so let me find that this is a new lab, meaning it wasn't in the included in the previous semester. And so in this lab, we're going to test the evolution or the amount of CO2. Let me share with you right quick. That your subjects or your lab mates or team will exhale based on a resting state and exercise state. At the same time, we would get things like heart rate and breathing rate so that we can match those with the amount of CO2. So how would you know how much CO2 is being exhaled? We'll use an indicator dye, and your subjects will blow into a beaker that has the indicators dye. So that was pretty straightforward and not too difficult. Then we get into things that are a little bit more complex, where we are using yeast and we're measuring the evolution of CO2 through the breakdown of sugars of yeast. There's two ways of doing this. One is with that vernier stuff that I showed you in the previous lab, obviously a different setup. If that setup does not work or it's not available, then we'll move on to a simpler version of this where we have yeast and only a few drops of particular sugar, and then we time to see any color changes in the yeast to sort of follow through in terms of whether or not the evolution of carbon dioxide increases with time or with the amount of solution of the prepared yeast. Now, this vernier lab, I don't have instructions for it, or you won't have instructions for it per se in the lab manual. There is a video that you need to watch so that you can see how it's done. All we have here is just a table for you to put your data. All right, so with that said, that's pretty much lab heads up. If you got anything from this, hopefully the takeaway was please read your labs before the actual lab. I would say at least a day, but if you don't have that time, at least look to it right before class. That may help. All right, questions before we move on to your papers now. Okay, I don't see questions, but I have a request. The request goes out to Victoria. Sorry to put you on the spot, Victoria, but you probably have given me the most or the clearest synopsis of these two papers so far. So I would encourage you, if you can, to sort of unmute yourself and give us just a brief synopsis of your perspective of these papers.
Wow, no
And
pressure.
I know, right? And thank you for doing this.
No worries. So the feedback feed forward paper was actually really interesting. And the long story short takeaway would be that your body can't start those feedback systems without knowing and predicting, and that would be the feed forward system. So think of it like, and we kind of went into the second paper too. Think about it like getting fit, and you're walking upstairs and you get winded. So your body goes, okay, well, I walked up 5 stairs and I got winded. So I need to be ready to provide more blood, more oxygen to the body the next time there's near stairs. So when I see stairs, I know I'm going to be ready to act. And that would be the feed forward system, that you have extra blood and oxygen just hanging out, ready to go, just in case you happen to go up 5 stairs. And then your body sets again to that new set point, and then maybe you can go up 10 stairs before you get winded, or whatever that might be. And then the second paper was about that idea of central command and the onset of exercise. Like, it takes a second for your body, for your brain to be able to tell your body when you are exercising, hey, this is what you're going to do to stay in homeostasis while you're working out, so that, your heart doesn't start beating 2000 beats per minute and you die of a heart attack. But in that moment that it takes before your central command takes over that homeostatic response, the feed forward system has to be ready to cover that gap. So between both papers, the idea is that feed forward is set by past feedback to prepare for the next maybe, and that it helps fill the gap before your brain or your central command can take over that homeostatic need.
Cool. Nice. Does anyone want to add anything? I have a few things to sort of expand on that, but literally that's it. These two papers are related. And they're related by the fact that they talk about this feed forward process. Now, if this is still confusing, because you are going to get questions on the exam about feed forward versus feedback loop mechanisms, either negative or positive, or what they refer to as closed loops and then open loops later on. Open loops are not a positive feedback, by the way. It's just their perspective on you need to know how to tweak these feedback mechanisms, and feed forward is part of that process that contributes to the idea of open loops. So one of the things that's interesting about both of these is that and I like the first paper a little bit more than the second one because of that chicken or the egg sort of pun that they put in there. And it's it's been shown that you can't really have one without the other. In other words, you cannot have feed forward without having the feedback loop mechanisms. And you can't have feedback loop mechanisms that are as efficient as they can be without the knowledge or the history behind feed forward. And so the thing we discussed in the previous class was just trying to define certain things. For example, what do they mean in the first paper in terms of sympathetic hemodynamic control? What were they talked about in general? Two examples of this in that paper, but one example sort of was talked about a lot more, or brought up a few more times, or a couple more times. What was that they were referring to in the paper as a constituent of this dynamic hemodynamic control, or control of blood pressure, basically? Does anyone remember that?
Restate that just for my clarity.
One more time. Yeah. So in that paper, the feedback and feed forward sympathetic hemodynamic control, they refer to a already innate system in the human body that already sort of accounts for that sympathetic control. And I was asking, what was the name of that? I guess if I say the word reflex, it might give it away. It was a reflex they referred to. But what was it? Which one was it? So remember that, as Victoria said earlier, right? There's a gap to be filled. When there are gaps in the body, in terms of sensory mechanisms and then the response to that, in order to shorten the time it takes for that to be addressed, you incorporate reflexes. And you can think of them as the simple ones, like the knee-jerk reflex, or this one that's more complex. So vasoconstriction was one thing they actually talked about, but then they kept going, they piggybacked on that, and they kept talking about the barrel reflex, or the onset of the barrel reflex in accordance to this vasoconstriction or the periphery effect. So the barrel reflex is that reflex found in the vascular system that helps maintain blood pressure. More so, it's dedicated to make sure that the perfusion of blood is adequate to the brain. And so one of the things they brought up in this paper was that, you know, when this reflex kicks in, at what point does feed forward sort of promote this reflex, or at what point does feed forward contribute to this reflex? And that's where they get into the chicken or the egg sort of discussion near the end. I think near the end they made another pun. I forget what they said, but it was something kind of funny. So that was one thing we talked about. And then in the second one, I have these figures there, not because you need to know them for any test, but just to give you the idea where the areas that they were referring to in terms of central command. Yes, it is the brain, but it's more so these parts of the brain that we sometimes don't even think of as such a, I guess, controlling unit, if you will. And these have to do with like the periaqueductal gray area or zone, which is essentially right in the lower drawing or figure there, and things like the globus pallidus and things of that nature. And the interesting part about all these areas is that in general, all of these areas have something to do with vasomotor movement, the smoothness of these movements, the balancing of these movements, and the fact that everything works in tandem, the signaling, right? When you include things like the internal capsule, the signaling between these is so efficient that we don't notice them working, and we only notice them working when there's an issue with them, like in Parkinson's disease. Someone that has severe Parkinson's disease, you ask them to stir a, well, you know, hypothetically, you can ask a person like that to stir a cup of coffee, and just to get the spoon into the cup of coffee takes an immense effort, right? They'll not convulse, but they'll shake, or they'll move their body, even their posture, in directions that we never thought would be ideal for the actual task. But this is what occurs when these areas are affected, right? You have a huge disconnect that we did not know could be there, because everything else was running smooth. And that's kind of the idea behind sort of taking these papers a little bit to the other level. All right, so with that said, I think we're almost out of time. Is there any other items you guys want to bring up before we call it a day? Right, so if you get questions, right, remember this, you'll get points for this once that assignment page is published. If you get questions, literally, they're going to come off of the stuff we just talked about. In fact, I can give you a heads up. One of the questions is going to be about, you know, what was one of the reflexes they looked at in that sympathetic hemodynamic control? It was barrel reflex, right? Yes, vasoconstricting was another, but it goes in tandem with that. And then another question for the central command one was, what do all these areas of central command have in common? Basically, the smooth control or smooth activation of muscle throughout the body, right? How they sort of feedback as muscles work together. So basically, they all work towards the same goal. There are specifics, but we're not going to get into that in this particular lecture. All right, I don't see any questions or any other. In future meetings, I hope to do less talking and have you all do a little bit more talking, although I think it went well today or tonight. 0% of the questions of or 0% of any articles will be on the exam unless I give you some fair warning. So, for example, let me escape out of here. Hold on. So, for example, let me see if I have that paper. Let me share here. Good question. I guess I can do the screen. So let's see. I think I have it here. Here it is. Oh, look at that. Perfect. Muscle memory. I didn't even know it was in that tab, but hopefully you see a green table. It says table 3. In fact, this table is in your IV fluids presentation. So for example, when we get a paper that's pretty technical like this one or somewhat technical, I may say, yes, make sure you look at this table or that table of the paper because you might get a question based on that or two. But for the first test, I don't foresee this. The papers, again, are there to expand our sort of connections in class and to address potential gaps in the topics we're talking about in terms of learning them and remembering the importance of these. Will I be extending? I don't think I'll be extending the quantitative paper. I think you have till tomorrow to turn it in, although it'll be called late. Tomorrow or Wednesday, one of those. There's the video in the IV fluids. I talk a little bit about how to approach some of these, and you also have the key. So there shouldn't be too much, hopefully, too much of delving too deep into this assignment. The idea of the assignment is to sort of refresh a little bit on the dimensional analysis spectrum of things, and also to give you a little bit of practice of potential problems you'll see on your exam. And I did say in lecture, you won't see problems as complex as these. And essentially, you will probably get something where all you have to do is plug and play. Of course, you have to know where this sort of setup is coming from. For example, the dissociation of particles, right, versus no dissociation of particles. That idea and topic I will talk about a little bit more in lecture. Other questions before we call it a night?
On some of the simpler dimensional analysis questions, you weren't expecting us to show our work, right? It didn't ask for it there, and
No,
not very much.
no, I'm not expecting. So those were the first ones on the boxes, right? Or something like that.
Yeah, like a good
Yeah.
first half of the page was just like
Right.
liters to milliliters, pounds to.
Exactly. No, that one I'm not expecting work. I mean, a lot of you probably can even use online calculators for that as well. But no, I'm not expecting work for that. Thank you for bringing that up. Other questions? All right. Ole, based on the feedback of silence, I'll call it an evening. This video should be ready in the same Zoom tab, hopefully within another 20 to 30 minutes. I will upload that page for your points as soon as I can. Your what's that thing called? Study guide for test 1. That page should also be up by tomorrow morning. So if you don't have any questions, I'll see you all tomorrow. Have a good night. And thanks for coming.
Have a good night.
Yep, thank you. See you tomorrow, right? Let's see.